Thank you again, everyone, for joining us. We're super happy to be sponsored by Amplify Ventures, who've provided all this food for us today. And today we're having Sarah Gurev present to us, who is beginning a Future House AI for Science independent postdoc fellowship, co-advised by Sergei Chenikov and Eric Schmidt. She recently completed her PhD in EECS at MIT in the Deborah Marks lab at Harvard, and she works on deep generative models of proteins with a focus on modeling viral and immune protein sequences and structures applied to viral evolution prediction, virus host interactions, and future-proof vaccine/antibody design. And we're super excited to hear what he has to say today.
Hi, everyone. Really excited to come talk to you all. Feel free to interrupt with questions at any time, and we'll try to leave some time at the end for additional questions as well. The goal of this presentation is, can we make early and accurate predictions of viral mutation effects? And let's consider a scenario where there might not be experiments yet to actually test these predictions or to use instead of model predictions. If we think about the SARS-CoV-2 pandemic, there's been a lot of data generated. So millions of SARS-CoV-2 genomes, so an unprecedented amount of sequencing. Anyone who's familiar with going into the PDB and looking at structures, and particularly antibody structures, will see that SARS-CoV-2 is also vastly overrepresented in terms of the number of antibody-antigen complexes there, and lots of experimental data. But we don't want to include any of that data in modeling for the moment. Instead, let's take a trip back to the start of the pandemic, pre-2020, and say what data could we have learned from then that could have taught us something about the SARS-CoV-2 pandemic. At that time, historical coronavirus sequences were available. And so our goal is, what can these evolutionary sequences teach us? So people have spent a lot of time trying to figure out how you can use deep learning models to learn constraints, meaning what makes a protein sequence functional from a set of related evolutionary sequences. So if we're talking about COVID, those would be pre-2020 coronavirus sequences. Think things like SARS-1, MERS, also common cold coronaviruses. So back in 2020, people would kind of freak out when their blood test said coronavirus on it, but that was actually mostly seasonal coronaviruses that were being picked up in these tests, because these are also coronaviruses, even if you don't really think of them in the same way as you think about COVID. But these all have kind of shared constraints and similar kind of functions and things they must do, and therefore we can learn about them to teach us something about SARS-CoV-2. How could we model this set of sequences? If we think about natural language modeling, there are a lot of parallels between there and the models we use for biology. So natural language, you might have a sentence using different characters or different words. DNA, we might model as a series of nucleotides, and a protein we might model as a series of amino acids. You can think about the tokens involved in each of these. So natural language processing, you might think words are actually a more informative token than individual letters, because there's a lot of meaning on an individual word that you don't quite have at a letter level. Similarly, DNA is sometimes modeled at a nucleotide level or a codon level, and proteins can be modeled at an amino acid level or sometimes k-mers as well. We're going to be focused in on tokens that are the protein alphabet. So 20 different amino acids that have different properties. And you can think about two different sequences that kind of fold to the same structure, but each individual amino acid in a given position is quite different. But you cannot just say swap two kind of positions with each other and expect to get a functional protein necessarily, even if they are sort of the equivalent amino acid. If you were to make a sequence alignment of these would both be at the same position, but purely swapping same positions in different sequences is kind of unlikely to work. You see kind of the clear clashes in this case. The problem we're trying to solve is, let's take a set of sequences that evolution has sampled. So doing its own experiment over these billions of years of evolution, it's taught us something about how to make a functional sequence, but we don't observe all possible functional sequences. So the goal is to take in stars the observed sequences and fill in the gaps between them of what other sequences are high probability and what other sequences are low probability. There are kind of two different ways we can think about doing this. One is an alignment-based model, thinking, let's just model those SARS-CoV-2 spike proteins, nothing about flu or hemagglutinin, HIV envelope, any of these other proteins at that moment. Just one model per protein family. The other way you can think about modeling is the sort of ChatGPT equivalent, where you're modeling not just, say, news text, but the whole set of the English language. And similarly here, you're modeling kind of millions and millions of protein sequences that are not specific to any viruses in general or even any one virus. In fact, when you look at these data sets are about 1% viral. And we'll ask how can these sort of like ChatGPT equivalent models, here called protein language models, teach us something about viruses in particular. And we're going to use both of these to do a task we call mutation effect prediction. Essentially, get a score for every amino acid at every position. What is the likelihood of that mutation, kind of given what we've learned from these evolutionary sequences? Starting first about alignment-based models, let's think about kind of what this data looks like. So you've probably seen multiple sequence alignments before, where these are now like into a fixed matrix of a specific length. There's like no insertions or deletions here. It's just all fixed length, and each position is sort of an equivalent part of the protein. And the idea here is that there should be kind of similar structure or similar function at given positions in this protein. So what does a model in a sequence alignment look like? Let's consider our pre-2020 SARS-CoV-2 alignment that was about 3,000 sequences. There are a couple different ways you can think about modeling these. The first is a site-independent model. So we have multiple sequence alignments. We're only going to look at one position at a time, this position I. And the question is going to be, at position I, what is the frequency of each amino acid at that position? And then you can, if you want to score a sequence with these position-specific substitution matrices, you can go and say, what is the probability or what fraction of the alignment is an A at the first position? What fraction of the alignment is a D at the second position? All the way to what fraction of the alignment is a T at the last position. And that can give you a score for a new sequence that you didn't see in your alignment. How likely is that sequence? Now, it might give you a reasonably good score, but there are kind of some problems with this. This is sort of my favorite example of epistasis, meaning sequence context matters. If we were to do the equivalent thing for names as we did for amino acid positions, the most common first name in the world might be Mohammed, the most common middle name in the world might be James, and the most common last name in the world might be Wang. However, the most likely complete name in the world is almost certainly not Mohammed James Wang. So similarly, you can't just treat positions in a multiple sequence alignment in a site-independent manner and expect that the context doesn't actually matter. We can go kind of one step more complex and look at pairwise interactions. So now instead of just modeling the frequencies at position I and having this position-specific amino acid frequencies, we can also look at pairs of positions at a time. So you have position I and position J. And the question is, how common is it to have K at position I and G at position J? So you're looking at all the pairwise frequencies and kind of tabulating those from your matrix. And so if we look at kind of our example here, that was kind of the positions we swapped out before. In one sense, we had the kind of the big amino acid and the small amino acid each being favorable, but when you swap them, they're unfavorable. And so by having a pairwise matrix, you can start to encode this sort of pairwise dependency. This is the sort of information that can help you get to 3D structures as well. And so when thinking about scoring a sequence, you now consider the product of each site factor as well as each pair factor as you build out a particular sequence. The way you can start to get structure out of this is to take a look at co-evolving sites, meaning when an amino acid at position I changes, does amino acid at J often change to compensate? And if it does so, and those are not near each other in sequence space, they're likely to be near each other in 3D contact space. So if this one mutates here to the smaller residue, we see the corresponding J here must mutate to a larger residue to compensate. And so this sort of coevolutionary signal that you can get out of a sequence alignment is part of what makes AlphaFold models do so well. Lastly, we can think not only of kind of explicit modeling of site and pair factors, but also move on to higher order interactions. So let's consider a variational autoencoder shown here. So here you kind of represent the input, one particular sequence at a time from your sequence alignment in a one-hot manner, and you kind of force that sequence down through a bottleneck, in a sense, making sure that you're not memorizing the sequences, and then back out to an output that's the same dimension size as the input. And the goal is to reconstruct the output sequence from the input. You're not explicitly using an alignment here, you're going one sequence at a time, but you have to use an alignment because this model is only specific to one particular protein family and is a fixed length. And so you can't even model insertions and deletions here. But because you're modeling the full sequences once, and then you have this encoder acting on the full input sequence, you are predicting mutation effects in the context of the full sequence. So when we use this variational autoencoder model to make predictions using our SARS-CoV-2 alignment, what we see is that models correlate quite well with experimental expression values in a deep mutational scan. So deep mutational scan is doing the same thing we were talking about doing in silico, where you have the effect of all mutations at each position, but now actually doing that experimentally, where we make a library of a lot of sequences. Yes.
Sequence alignment on a whole genome or then specific protein?
For the most part, we're talking about protein language models here, which are at a protein level. Yeah, you can also do this at a genome level. If you're talking about making an alignment, nucleotide level alignments are actually a lot harder to do than amino acid alignments. This is part of the reason why like RNA structure predictions are a harder problem for multiple other reasons as well. So sometimes when people do genome models, they actually treat them as sets of proteins and do like all the different proteins together because there kind of is more information that we can learn at the moment from these protein level models, but kind of genome level models are sort of starting to get better in different ways. So yeah, so we have our model predictions and we have these experimental predictions, which are these deep mutational scans that actually measure expression levels for each mutation. And this here is each mutation to the receptor binding domain of the SARS-CoV-2 spike protein. And you see they correlate pretty well. What's interesting is the part of this scatter plot that's off the diagonal, essentially where the model says these mutations are not functional, but the experiment says they are actually sites, so the receptor binding domain is in contact with the rest of the spike protein. And the experiment only modeled the RBD alone, so it missed the context of what are the constraints relevant to the full spike. But the model picks up on those because it is in fact modeling the full SARS-CoV-2 spike sequence. And so kind of this is just one example of showing kind of what we can learn sometimes differently by experiments versus models. One thing that we did was combined what we can learn from evolutionary sequences with some structural and biophysical information in order to predict antibody escape, meaning what mutations are going to make it so the SARS-CoV-2 antibodies that we make no longer are able to bind. So that's why you all had to get new boosters for different SARS-CoV-2 variants, is the virus evolves such that the later versions of the virus no longer bind the earlier antibodies you made to an earlier variant. And so you have to update the antibodies you make to be newer and newer variants. This then allowed us to say, what kind of fraction of pandemic variants could we have predicted, kind of using only pre-2020 information, and how will do experiments predict this? And so if we look at another set of deep mutational scans, this time not just on expression, but actually looking at binding to pandemic antibodies, we can say what fraction of mutations that these experiments said will lead to antibody escape, meaning leading to mutations that will make you no longer bind these pandemic antibodies. Given the experiments actually tested binding to pandemic antibodies, we saw that these experiments, available 10 months into the pandemic, were able to predict around 30% of the mutations that actually arose up until 2022. Later experiments using more and kind of more up-to-date antibodies from the pandemic were able to predict closer to 40% of the mutations that actually arose. And we saw, yes.
Oh, just by earlier and later, they were using data that was available These experiments were kind of run in the same.
So no, these are so it's kind of two different like one set of papers that's earlier. This is like the very first antibody escape DMS that was done, and it was available 10 months in the pandemic. And then 17 months in the pandemic is later versions of it's basically the same exact experiment, but with kind of more antibodies. And so they just updated the antibodies they were testing against. And so these take time to do because like both the experiments take a lot of time to do, and you actually require the actual patient like antibodies to be up to date. And then we talk about our model. We called this EVEScape for predicting antibody escape using these evolutionary sequences. Using information only from the start of the pandemic was sort of just as good or better than these deep mutational scans at predicting the actual mutations that arose during the pandemic. No, you never expect this to get to all the way to 100% because a lot of the mutations that arise in the pandemic have nothing to do with antibody escape. They either just come along for the ride with other mutations, like if something else is important and it kind of just like also mutates at the same time, that one might take off too. Or especially at the very start of the pandemic, why it takes kind of time for this to increase is because the early mutations were not antibody escape. Noticeably, we didn't have pandemic antibodies until we had a vaccine or you had your first infection, but really more about human adaptation because SARS-CoV-2 was a spillover from an animal into a human. And so these earlier mutations kind of had nothing to do with escape. And so this kind of goes to show these computational models could have been very important for predicting the mutations that would arise, but doing it at day zero, with the hopes that maybe you could make a vaccine from the start of the pandemic that could protect you against kind of what you expect to see later in the pandemic. But yeah, as we noted, this was available at the very start. We're also starting to do similar work for flu. So H5N1 is the avian influenza. It's the reason why your eggs were so expensive and scarce for a while. It's because H5N1's been circulating in poultry. And then H1N1 is one of the seasonal flu vaccines that you get every year. And also, when people talk about being like a good year for the vaccine or a bad year for the vaccine, that means sometimes the vaccine's a better match for the variants that are actually occurring this year. And sometimes we pick kind of a bad match where the virus ends up kind of evolving in a way that we kind of didn't expect when we had to produce the vaccine 6 months before flu season. And so every year it's this problem of we have to start making these vaccines 6 months before flu season, but we want to protect against the flu that will actually be circulating during flu season. And sometimes we're good at guessing and sometimes we're bad at guessing. And that's kind of an interesting avenue of where these predictive models could help. And here we show a similar thing, EVE, so that these evolutionary sequence models do better than the deep mutational scans, the DMSs, at predicting the evolution that actually happens during these outbreaks. A second way to look at these models is ask a different question. Can these computational models design versions of the virus that look like real future versions of the virus in terms of their behavior with respect to antibody binding? Meaning you all got probably a couple different versions of the SARS-CoV-2 booster vaccine. And when people made the booster, they had to choose which variant they wanted to include. If the goal of choosing a variant that would kind of continue to be useful more than a year into the future. But when they wanted to experimentally test to what extent they think that vaccine would be protective, they would test it against existing circulating variants. What we propose is that you should test your vaccine not just against currently circulating variants, but also against likely future versions of the virus in order to say, do you expect this vaccine to continue to protect you into the future? And so the goal here is to kind of make future variants that look, in terms of antibody escape, similar to actual variants. And so antibody escape is considered with respect to patient antibodies from a number of different infection and vaccination histories that are sort of representative of the population. Essentially convalescent with an original wild type or Delta virus, different vaccines, boosters, or breakthrough infections. So well representative of kind of infection vaccination histories. And this here is an antigenic cartography plot. It'll be somewhat similar to like a PCA, where points together behave similarly in terms of the sort of 9-dimensional antibody escape data, and points further apart are more dissimilar. If you look at actual pandemic variants, colored here over time, the variants earlier in the pandemic clustered together, and then they get kind of further apart later on. Interestingly, this is SARS-1, meaning SARS-1 is as different from kind of the original SARS-2 as these kind of later SARS-2 variants are. So we are now further away in kind of antibody space than SARS-1 was to the original SARS-2. If we think about, so these circles were our natural variants. These triangles are designs where you train on information up until one variant and ask what design variants using about 5 to 10 mutations, do those actually look like true later variants? And so here we say they look like variants that actually arose 12 months later, 3 months later, 8 months later, 9 months later, and again 12 months later into the pandemic. Meaning here, if you design on the XBB background using information up until this point in time, it ends up looking like variants that actually arose 12 months later. Not necessarily, but sometimes in terms of the exact sequence, but actually in terms of the behavior with respect to kind of patient antibodies. Meaning if you wanted to pick a vaccine at this point in time, you should use this panel of variants to actually test your vaccine and say, will this vaccine be future protective a year from now? So our goal is that we could be able to use these models to aid in surveillance, to evaluate future protection of vaccines, and also for vaccine design itself. So we've been talking here kind of about alignment-based models, and I showed you one particular example of an alignment we made, but one question is how to actually choose the best alignment, assuming we didn't have experimental data to help us say this is the alignment that we want to use. Before, people would take a protein sequence, query some different sequence databases, make a multiple sequence alignment, and then just choose the deepest alignment, thinking if there's more sequences there, there's probably more information, and so the model is going to be better. And so sometimes this works. So here's an example of a deep mutational scan on a polymerase protein, whereas the alignment gets deeper, so there's more sequences, performance, the Spearman against a particular deep mutational scan gets better. So that's sort of what you would expect. But we saw deeper alignments are not always better. If we look here at this protease, we see as you increase alignment depth, model performance actually decreases. So we wanted to say, when is this going to be true? And so we made a virus-specific experimental data set to look at these best modeling practices. So we collected and standardized 45 different viral deep mutational scans across many different viral families and across many different assay types. And so then we could ask the question, would something like alignment relevance be a better predictor of model performance? Meaning, let's take a look at this Coxsackie alignments and why, when you're at these very deep alignments here, the model actually gets worse. So if we look at kind of one of these alignments and look at what fraction of the sequences are at what percent identity to the original query sequence, the one that you did the deep mutational scan on, and you saw that less than 0.01 of this alignment is within 10% identity to the query sequence, and most of the alignment is less than 25% identity. This is in contrast here to the shallower alignment, where almost 15% of the alignment was within 90% identity to the query, meaning like all of these sequences are probably in the deeper alignment, just kind of overshadowed by a large number of sequences that are highly unrelevant. And so you're kind of losing the signal that was here by kind of overwhelming it with these sort of less relevant sequences. And so we say if we move from a regime that's kind of what's commonly done of using a deeper alignment and instead look at something like what fraction of the sequences have more than 90% identity to the target, you can see that you can get better performance when you're using relevance instead to select your alignment. So our question was, we've now created this benchmark to ask questions about what model parameters will help you or not. And we've taken a good look at alignment-based models for viruses, which is what we use when we had previously done this SARS-CoV-2 work and this vaccine evaluation work. But kind of protein language models have become kind of very important in other protein design fields. And so we want to ask the question, what if you move out of this regime of a single alignment of one protein family and instead want to look kind of beyond that? So do protein language models actually work for viruses? So there's been a big rise in large language models for proteins. Here's a kind of a number of different examples with ProGen models, ESM models. And the way these are trained are they're trained with self-supervision, meaning there's no labels. So this is a protein and this is the expression level of that protein, and we're training on labeled data. You're only training on the sequence itself with sort of the assumption that this was a sequence that was an observed sequence and therefore it is functional. So it's self-supervised by that sense. And we're not aligning these sequences. So when people went and trained ChatGPT, they weren't saying, this is the sequence and I've diagrammed that this part's the noun and this part's the verb and all that. So we're no longer aligning our sequences. We're just kind of taking as input either kind of an autoregressive sequence where we have kind of the first part of a sentence or the first part of a protein, and we try and predict the next token, or we have a kind of a masked regime where we have the full sequence or the full sentence, and you have some portion of that sentence or sequence masked. And the problem here is at these masked sites, what are the amino acid at those sites? And so here you're just saying to say you have 1 to 20 amino acids, which one goes here, and here we've got about 15% of the protein masked, and we're trying to kind of fill in the blanks at those masked sites. So the first thing we did was take a look at how much data set influences how well these protein language models do. And we looked at Protein Gym, which is a different set of mutational scanning benchmarks that has some viruses, but also has more than 200 mutational scans on other taxa that aren't viruses. And so what we see is that each dot here is a different protein language model, and it's colored here by the data set that protein language model was trained on. So mostly UniRef or subsets thereof. And so you can see when you're talking about all taxa that it really doesn't matter what data set your model was trained on. You're sort of doing basically equivalently well. Some of the best models were trained on UniRef 90, so this light blue. But when you're in your virus regime, it really matters what data set you train on. Like you're basically only going to do well for viruses if you're basically a UniRef 100 model. And notably, this line here is the site-independent model that we talked about before. And so a lot of PLMs are doing better than a site-independent alignment-based model for non-viruses. But in the viruses, all but these sort of like handful of UniRef 100 models are kind of doing worse for them than just the simplest site-independent model. And so kind of what's going on here? So if we talk about these sequence databases like UniRef is just a set of sort of non-redundant protein sequences that have been collated together in a database. The problem is that these sequences are not independently distributed. And so there's lots of clusters here with kind of more similar sequences, and then there's some like standalone points that kind of have nothing similar to it. And so in order to avoid overfitting your model to these kind of very well represented clusters, what people do is they cluster their data and only train on one representative sequence per 90% sequence identity cluster, meaning no two sequences that you're training on should be within 90% sequence identity to one another. So this allows you to kind of avoid overfitting to these large clusters. And so as a result of this is what most protein language models have been trained on, either UniRef 90, so one sequence per 90% identity cluster, or UniRef 50, which goes even smaller to one sequence per 50% identity cluster. But let's take a look at kind of what's actually in these data sets. In general, viral sequences are only kind of less than 1% of their training data set. So you can look at UniRef 100, UniRef 90, UniRef 50. PDB, which is like the protein data bank, which has relatively few resolved structures. And then AF2DB, which is the AlphaFold database. This version here is essentially AlphaFolding almost everything in UniRef 50, but not quite everything, and also explicitly removing viruses, besides kind of some prophages, which sneak in because, yes.
On the previous slide, sorry, the similarities are computed how similarity between two.
So it's like you have two protein sequences in here. It's like line by line, if you align these sequences, what fraction of positions are the same. And so yeah, so you can see if you go from UniRef 100 to UniRef 90, if you're looking at the total size, you're going from like about 450 to 200, so a little less than 50%. If you look at like one particular viral protein, here the paramyxovirus F protein, you go from like 6,000 to 600. So that's only about 10%. And yeah, so if you look here, there's also, you see there's like no examples of these important viral proteins and like AFDB. And some of these viral proteins are really overrepresented in the PDB. Like there's a lot of influenzas or a lot of SARS-CoV-2 proteins and some that are quite small. If we kind of look systematically, we took a question of how, for a bunch of different viral and non-viral proteins, what is the number of sequences that are in a sequence alignment if you did this kind of sequence search in UNIF 100? And if you did this clustering at 90% identity, how many effective numbers of sequences would there be? And so if you look for basically everything besides viruses, you find pretty consistently for every 8 UNIF 100 sequences, there would be 1 UNIF 90 sequence. So follows quite well along this diagonal here. But you see for viruses, there was a lot of viruses way over here, meaning 550 UNIF 100 sequences to 1 UNIF 90 sequence. So you see that viral sequences are kind of disproportionately affected by sequence clustering. The difference being, besides HIV, which is just like an absurdly diverse virus, and then phages. So phages are viruses that infect bacteria, and these are often actually double-stranded DNA viruses. So you can have viruses that are made out of RNA, or you can have viruses that are made out of DNA. And viruses made out of RNA mutate at orders of magnitude faster than sort of like any other organism out there, where double-stranded DNA viruses are more similar mutation rates to kind of these other organisms. And so viruses, because they mutate so fast, they actually kind of quite quickly in evolutionary time escape the limits of homology detection, meaning by like if you try to look at two sequences that are kind of similarly diverged in evolutionary time between the viruses and bacteria, the virus ones would have kind of no longer detectable sequence similarity, where the bacteria kind of might still be 50% identical to one another. And so that makes it so that you have much fewer relevant sequences when you're querying for any one sequence. And why, when you look at UNIREF 90, you have this giant loss in sequences for viruses that you don't see in these other cases. So this has made it so the PLMs where it's useful to train on UNIREF 50 or UNIREF 9 or 90 for other things. For viruses, it's like really bad. Something else we looked at was how does the number of parameters impact model performance? And so people showed kind of before that there was this interesting plateau as you increase the number of parameters. How well does your performance, essentially your Spearman correlation with each of these different deep mutational scans matter? And this is looking at all texts and not just viruses. And so you see kind of a clear plateau that happens here around like 10 to the 8 parameters. And you see kind of like both autoregressive and math language modeling models are doing basically equivalently well here. For viruses, on the other hand, you don't see any kind of plateau yet, meaning kind of bigger models are continuing to help even as you get to these very large 10 to the 10 model sizes. What's likely happening here is because of the lack of training data relevant to viruses, the bigger models actually help you generalize from the non-viral information that's further away to the viruses. And so they kind of help you compensate for less sequencing data. It's also interesting that you can see that basically mostly the UNIRF 100 and UNIRF 90 plus BFD models, like the only ones that were trained using this data set were the autoregressive models. And so you actually see the autoregressive models at the moment are doing better than math language modeling models for viruses at the moment. So why don't we just train always on these larger sequence data sets? So why isn't everything on UNIRF 100? Training's time and resource intensive, and this clustering has historically improved performance for non-viruses. So if we don't want to solve this problem by kind of training on more sequence data sets, how could we potentially learn from other data modalities? So something else we saw was that structural information could help compensate for low sequence diversity. So here now each point is one of those 45 deep mutational scans. And we have our Spearman correlation between a different model and the DMS. And we chose 1 unit of 90 example, 1 unit of 100 example, and then one protein language model that's trained not just on protein amino acid sequences, but actually in a structural aware manner. Meaning when we talk about the token being an amino acid before, now the token for a given protein is an amino acid concatenated with some local structural information. So it gives some sense of what the local structural space looks like around that amino acid. And it's trained here now on the PDB and AlphaFold DB. And so you can see that for viruses, the structural information is helping to compensate for relatively low sequence information, and that this is actually doing quite well for viruses. And you can see that it's doing particularly well here in some like stability type assays. So if you're talking about protein stability, not necessarily a particular function, structure you'd expect might be particularly helpful because that will really tell you, like, does this seem to be a stable protein or not? So why might this be helping? So if you're talking about like one particular virus, like a Coxsackie protease, there can be sequences that are less than 30% identity, so or even further than that are very high structural homology. So here is an example of a human serine protease domain within RMSD 3.6 angstroms. So essentially, if you think about the embedding space that the protein language model is in, there might be two sequences that are here both proteases, but are very different in sequence space. So they're kind of in two very different parts of this embedding landscape in a sequence-only protein language model. Now, if we think about a structural aware protein language model that learn from structure, these sequences are more likely to be near each other in this embedding landscape and actually learn from the sequence dissimilar, the structural similar information when it's trying to make predictions. And so these structural models can learn from these remote homologs if the structural information is there to tell you that those are kind of relevant, interesting sequences to be looking at. Yeah, and what we see here is stability assays, and it kind of helps you particularly for these structural information. Yes.
Any ideas on why stability is more of a structure than physics?
So I mean, if you think about if you had a structure in front of you, things you might be able to like tell about it. Like if you have like a structure, it can be quite easy to say this looks like it is folded properly and there's like bonds in kind of like the right places. There's not like some floppy thing all the way over here. Like you can like learn a lot of things from a structure. You can also tell that like if it seems like it's folding okay, it's probably expressing. So like fitness is sort of a step above just expressing or a step above just folding properly. You have to like have folded properly and expressed and actually perform the binding function or whatever other function you need it to happen. And so that's where the evolutionary sequences is really going to teach you something that a structure is not. So another question we tried to ask is, like we had before, and we're trying to say which alignment is the best alignment, meaning is a deeper alignment better or is a more relevant alignment better when we didn't have experiments to help us choose the alignment. We want to be able to say this generally. How do we know which model we should be using for a particular virus? Or in fact, we can trust models at all for that virus. Let's say on day 0 of a pandemic, there's a new virus, there's no experimental data yet. I'd like to use my model to make predictions to help inform vaccine designs. Do I trust this model? And so we created confidence metrics, either for protein language models or alignment-based models, and showed that they correlate well with the Spearmans, with the deep mutational scans for these different viral proteins. And so when we look at an alignment, we'll use a measure of sequence diversity. So how different are the sequences that you're training on when we look at the sequences that are highly relevant, so within 90% identity. The thought that the more sequences there, kind of the more information that you have to learn from. And we use a pseudo-perplexity method for protein language model reliability. In a sense, how uncertain is the model about an output? If it says at a given position that all amino acids are equally likely, and it says this throughout the entire protein, it probably has no concept of that protein, it's not going to be any good at making predictions. Whereas if it's usually only saying like 2 or 3 different amino acids at a given position on average, then it likely has a better knowledge of what's going on for that protein. And so what we showed was these reliability metrics can predict performance. So we call this approach, where we are able to do accurate reliability estimation, evolutionary variant effect prediction with reliability estimation, or EVEREST. What this allows us to do is actually say for viruses we don't have experimental data for, namely the WHO priority viruses, which ones of these could we actually make accurate predictions for using the models as they are now. So we look at EVE, the variational autoencoder in the MSA case, in the alignment-based case, and we look at SA-prot in the PLM case. And colored here are the different viral families that are covered by these 40 WHO viruses. And we see that only a scarce few, mostly influenza or HIV, are good enough for both protein language models and alignment-based models. A lot more are good enough right now with alignment-based models. And then there's many, many that still we wouldn't trust these models for. So it would be about below 0.4 Spearman correlation if you were to do a deep mutational scan. Meaning there's not no information there, they're just less accurate than these guys up here. So our goal is that we could be able to use this to continue to accelerate pandemic preparedness and also for biosecurity assessment. So people often say, when I talk about doing AI for viruses, like, is this a biosecurity threat? And this is one way to assess whether a new model actually might be a biosecurity threat, and should you consider different kind of access protections, and we can use this sort of approach to evaluate them. So we've released all the in silico deep mutational scans, so the predictions for each mutation for all these 40 WHO priority viruses on our GitHub. So many of these don't have a deep mutational scan yet, so they're kind of the most comprehensive mutation effect predictions out there right now. One example of what this looks like is for the Zika envelope protein. So this is what kind of a mutation map would look like, where red is the very likely mutations and blue is the very unlikely mutations at each given position. We have the full protein, but just showing you a small number of positions so that you can actually read this. The model we use here is a new hybrid model that we make that's essentially a fancy average of SA-prot and EVE. So EVE being like the best alignment-based model and SA-prot being the best PLM. And so if you see here, this area that's very blue actually corresponds to a known conserved fusion loop. And this area here that's very red corresponds to an area that's a known variable glycan loop. And you can look at this on the structure as well, where this is kind of the average mutation effect at each position. And you can see some clear areas that are highly mutable or highly unmutable. So if you're thinking about designing a therapeutic, you might want to target an area of this protein structure that's a lot more conserved and so less likely to mutate in the future. So overall, we've been asking the question of what can evolution teach us, and can we learn from functional examples of protein sequences, constraints that are relevant to evolution and that could help us with vaccine design or other sorts of problems. And so our goal is to be able to use these to, again, aid surveillance, help evaluate our future protectiveness of vaccines. And we're now moving into an era where we're starting to do some actual vaccine designs itself using these sorts of approaches. So with that, I'd like to thank lots of people in the Marks lab, especially Noor, who co-led a lot of this work, as well as our experimental collaborators in Lemieux, Lubin, and Seaman labs, who helped particularly for the antibody experiments and infectivity experiments that went along with the vaccine evaluation project. And so here are some of our papers. If you'd like to take a look at any of them, particularly our work on antibody escape prediction, vaccine evaluation, and our new work on EVEREST. And so with that, thank you very much. Happy to take any questions, and always feel free to reach out if you're interested in machine learning for viruses and immune proteins. Great, thank you. Questions?
What do you think led to the performance of these? Like what architectural details allowed you to model like what you thought the effective variance would be?
Yeah, so our goal with Evescape, so that's the model where we took the VIE on the evolutionary related coronavirus sequences and added in some structural and biophysical features that were relevant to antibody escape. So logic there was if we take a look at the sequences in our alignment, they were mostly bat or pig coronavirus sequences. They weren't sequences that had already been circulating in humans and under an antibody escape response. And so how do you combine kind of things that will lead to increased fitness, and so just being a functional virus, and add in like a bias towards things that will actually matter for antibodies. And so in order to escape, we say you have to be a functional protein, so that you can learn that from the evolutionary sequences. You have to be an area where antibodies bind, so you can look at a structure and judge where antibodies are likely to bind. And you have to be a mutation that's actually different enough than the original wild type amino acid there that an antibody that previously was able to bind is no longer able to bind. If you think about what you're learning from evolutionary sequences, you're generally learning like substitutability, thinking like a blossom matrix, where I'm likely to substitute for another amino acid at my same site if I'm similar in terms of charge and hydrophobicity and size. But in terms of antibody escape, it's actually the complete opposite, where it's not the more similar you are, the better. It's the more dissimilar you are, the better. And so we just came up with this framework where you could learn from these evolutionary sequences and add on these kind of bias terms to the part that's relevant for antibody escape. Any other questions?
First, I'm on the core bio side and machine learning, and I think your presentation was really accessible to me.
Great.
So I do work on like bacteriophages, and you mentioned them a few times here. I was wondering if you had thought about applying the same kind of, I don't know, framework to bacteriophages, because people are looking at using the bacteriophages to, either kill specific bacteria or like try to deliver genes to the bacteria.
Mm-hmm.
In those same contexts, it's like you'll have a tail fiber that's binding, that's protein that's binding to a protein that's on the surface of the bacteria.
Mm-hmm.
I think you kind of flip the dichotomy, where in that context, the antibody is more of like the tail fiber on the phage, and I don't use the antibody words, but you flip it around. And so I was just wondering if you had thought about, maybe applying it in those contexts of phages and bacterial hosts.
Yeah, I think it would be really interesting. There's a lot of ways where this type of model can start to be used for phages. One is just purely like functional annotation. There's a lot of bacteriophages that we have like no idea like which part of a phage they are. And so sort of protein language models can sort of be used to annotate things with unknown function by just saying, are in embedding space, like, are you close? Are you similar to already annotated bacteriophages proteins? And so that's one place they're starting to be used a lot, particularly like structural aware protein language models can also like be helpful for that. Or you could, there are similar types of models that can avoid having to like essentially fold all the different proteins. Like we had talked about how structural signal stays a lot longer than sequence signal. And so people use kind of protein language model approaches to come up with a representation of the local structural space of a phage, and then look at that structural comparison to annotate. So that's like one way they've started to be used for phages. Just being able to predict phage evolution generally, I think would be super interesting. And also phage like receptor specificity, like you can use some other types of protein design models to start to get at host predictions for phages. Yes.
I have two questions. One is on EVEREST.
Yes.
The metric to measure uncertainty from the model said it was like this pseudo perplexity.
Yeah.
Wait, so what is that?
So pseudo perplexity is essentially you go position by position in your sequence, and you say like how many amino acids on average are you kind of predicting as sort of like equally likely at that position. And so if everything's like equally likely that's completely flat, then you're essentially going to be predicting like 20 possible things at that position. Where if it's the prediction landscape for any position is like not flat, meaning there's like only like 2 or 3 things at that position, then that's going to be like a much like higher score And so it's essentially on average, if you go position by position, how many things are you saying are likely at that position?
Okay, so kind of like an entropy.
Yes, exactly.
And then on the graph where you showed the World Health Organization, like
Mhm.
viruses, are there any of those that are like, I don't know, something on the right-hand side that's like especially exciting, and then something on the left-hand side that's like especially concerning, or are all of these like concerning viruses?
They're all on the WHO priority list because they would be really bad if they were a pandemic, and also they're relatively likely to be a pandemic compared to other viruses out there. Something I think is interesting, like this here is Lassa virus. So it's interesting that it's doing like quite well for alignment based and then so badly for the protein language model. I think it's because like there is a lot of circulation at the moment, at least like amongst like rodent reservoirs. It's not doing a lot of like human to human transmission yet, but it's like doing rodent to human. And like we've been doing a lot of sequencing of those rodents. And so it's interesting to think about how those like somewhat related sequences in those rodents can help us teach something about this virus and maybe help us make therapeutics that would actually protect us against the different versions of that virus is something I'm like interested in studying. But yeah, there's other arena viruses that are like all the way over here where we essentially don't know anything about and really telling us that we should be like sequencing a lot more of the related viruses over here. Yes,
Actually, have I think so. One is for the training.
yes.
So you said not necessarily having more data necessarily improves the result more. So because you need ref, I think the name was the protein language model seems to put all the proteins information in
Mhm.
like for more like a general purpose.
Yeah.
So it's more like radical, but why not just focus on virus, the training the data, so it can really fed into that virus rather than just dumping other viruses data to make it like an artifacts.
So you're talking about like why not train a viral protein language model?
Specialized.
Yeah. That's actually yeah, so that's something that we're actually particularly interested in doing, something I'm working with Sarah on doing, who's an undergrad who's been working in the Mark's
I
lab.
guess you're speculating that's maybe prover.
Yeah, that's the hypothesis, that we should train on viruses or even not just viruses, but things that are similar to viruses. And that you take what we've learned from this analysis to kind of make the optimal model, in this kind of case. Because agreed, there's a lot of kind of problems at the moment with these general purpose models that don't apply well, especially to viruses.
When the mindset is so it seems like you're pushing your research into like the vaccine design.
Mm-hmm.
So now we have like bivalent.
Mm-hmm.
But speaking about multivalent, maybe future development and valent is something. So it's like so you have to pick like multiple strains, but how do you kind of predict which strains are different, not so similar to each other, so that you can pick up the right ones?
Yeah, so we kind of think about this vaccine design question in like a somewhat similar way as we think about this antibody escape question, meaning the thing that's going to cause antibody escape is the thing that's most dissimilar to the currently circulating strains. But you also think about two vaccine strains. If you wanted to pick two vaccine strains that are far apart, it's essentially the same type of model, but just like you choose one vaccine as your starting point and you choose the other one to be like most antigenically dissimilar. So we use this sort of approach for like a multivalent vaccine, looking at could we optimize a nanoparticle vaccine? So a whole different type of vaccine modality. So for SARS-CoV-2, everyone got an mRNA vaccine for the most part. And so for the mRNA vaccine, it's sort of like each protein kind of separate. If you have a nanoparticle vaccine, it actually has a kind of a common scaffold. And then you have a lot of different versions of this protein on top of the scaffold, so kind of all connected together. And so if you kind of have that, you can think about optimizing if you have kind of multiple different proteins on the scaffold, how do you make them different and how do you optimize where they are different? So put mutations in there that make it so that only certain parts of the protein is conserved. And you think about the parts that are conserved if you have different things on the scaffold being the parts you're kind of more likely to make an antibody response to. Especially antibodies are bivalent, they have kind of two arms, and those two arms are the same. And so an antibody is going to bind with avidity, like with both of its arms at once, to parts that are conserved between two different proteins next to each other on the scaffold. So we thought about how to like optimize the proteins that you would put on a nanoparticle scaffold as one way of doing vaccine design. We've also, talking about for bird flu, thought about, at the moment, this is kind of currently circulating, mostly not in humans. Could we predict where we think the virus is going to go and how to make a vaccine that would protect us, not just against kind of the version that's circulating now, not in humans, but against kind of the likely outbreak sequence. And so can you make targeted mutations to this virus that will help protect you against the likely version of the virus that would actually be the outbreak virus?
If you don't mind.
Yeah, go for it.
So most of the person when something like H5 and all, so it's becoming usually,
Yes.
effective. So I mean, the core of all the evil, I would say, will be the footfish, right? So happening in the avian flu, right?
Mhm.
Chicken. So I know it's kind of a money issue, but are we trying to kind of vaccinize the chickens,
I think
just, trying to touch the center of it?
I think that's ideal. There's kind of the whole work on like veterinary vaccines is sort of kept completely separate to the work being done, I feel like, on like human developed vaccines. But I think it's really important that, especially for these viruses, that for it might make the jump into humans, that we actually do vaccinate animals against them. And there's a lot of viruses that still don't have like a good vaccine that I think would be really important to do. And so yeah, there's a lot more work to be done there. At the moment, one of the things that needs to be done, just like to get a good bird flu vaccine that's being worked on, is just like purely just stabilizing the viral antigen. And so when people got like a SARS-CoV-2 vaccine, it wasn't purely the SARS-CoV-2 spike protein. They had to add some mutations to stabilize the conformation. So SARS-CoV-2 spikes are trimers, meaning there's kind of 3 copies of it. And then there's one particular part, the receptor binding domain, that can kind of be up or down. And so when it's up, it's binding ACE2. And when it's down, it's not binding ACE2. And so that's kind of one conformational change it makes. It also makes an even bigger conformational change when it fuses with the host cell. So it's kind of in the shape that lets the receptor binding domain kind of bind or not bind in the pre-fusion state. And then post-fusion, it makes this giant different conformational shift that kind of elongates parts of it and like looks completely different. And the parts that's exposed to antibodies in this like pre-fusion state is like totally not the part that's exposed in a post-fusion state. If you think about after the virus fuses to the cell, you're not really making antibodies at that point, or those antibodies aren't actually going to be useful to prevent infection. And so you really only want to vaccinate against the pre-fusion conformation. And so people are trying to do is to stabilize the virus into a pre-fusion conformation for the vaccine. And so that sort of thing, that same sort of pre-fusion stabilization is relevant for flu. And so that's also part of what's going into like a good influenza flu vaccine design that would be like an mRNA type vaccine. At the moment, the seasonal like flu vaccines you get right now are not they're sort of like single protein or mRNA vaccines. They're actually like egg or cell based, where you're actually kind of sort of like getting a whole virus, then like chopped apart into like bits of it. And that's just like a whole different type of vaccine where you don't need to do that kind of stabilization. But ideally, we would have the same sort of like mRNA vaccine that we have for COVID.
So like how difficult is it to collect data on these like the viral antigens? Like most of the viral structure databases that have been released are all like computationally predicted. Is the main variant like the determining the structure of it, like through like extraterrestrial?
Yeah, so if we just look at how many structures are out there experimentally determined, the scale of structural databases experimentally determined is just much, much smaller than the scale of sequences that we have. And so in order to like learn about structure, we have been overly relying on computationally predicted structures. So for one, viruses were explicitly removed from these computationally predicted structure databases, and only in the last year is starting to be predicted in bulk. For another, because these structure predictions like use sequence alignments in order to help make them, and there are fewer relevant sequences. The same way that this makes them worse at mutation effect prediction protein language models, the structural predictions are actually worse for viruses than they are for non-viruses. So things like AlphaFold have lower PLDDT, meaning like predictive confidences for viruses than they do for non-viruses. So that's like one bottleneck. Also, just like in terms of doing comprehensive prediction, like a demutational scan, demutational scans are like exceedingly time intensive. And so we sort of like never expect that we'll have a demutational scan, meaning a prediction for every mutation for all of these viruses. The Bloom lab was doing kind of a number of different viruses, kind of like that are important ones, but I sort of just like never expect that that's going to be any like lab's priority to release a demutational scan on everything. And so it's going to be like if you want to know about kind of any position mutation, it'll be kind of important to do that computationally. Also, like demutational scans, you can't do like all combinations of mutations. Like if you quickly try to do like all doubles, even all doubles would be like way too many to like test or to even make a library for. And so moving beyond singles to like higher order mutations, you're going to need a computational model to kind of predict the effect of any combination.
I think.
Oh.
So are proteins that are like multimers, so like, you know, dimers, trimers, you mentioned spike proteins,
Mhm.
right? Are those harder to, yeah, predict things about, I guess, in systems are than, or is there like alignment based better, or is like a structure based be better, or is it?
So at the moment, like structure-based models would probably just like almost entirely miss any constraints that come from the multimerization because for the most part, when you're folding them, you're only folding them as monomers. And so it's just missing that entirely. Sequence-based information, like theoretically, like the constraints underlying the within the monomer and between monomers are both sort of like equally encoded within the sequence constraints. And so you expect that shouldn't matter there.
If there are no other questions for now, maybe we can, and then Sarah will stick around for any questions if you have anything. Thank you so much for coming and thank you, Sarah, for speaking.
Great, thank you.